In complete darkness, 600 meters beneath the surface of the Indian Ocean, something impossibly long drifts through the void. At first, the scientists operating the submersible Sebastian think their cameras have malfunctioned. The creature before them spirals through the water like a galactic whirlpool made of living tissue. Its outer ring spanning 47 meters, each segment glowing with bioluminescent fire. It is longer than a blue whale, longer than most buildings are tall, and yet it moves with such ethereal grace that it seems less like an animal and more like a nightmare sculpted from smoke and starlight. Dr. Nerita Wilson, the marine biologist leading the expedition, watches the monitors in stunned silence. She has spent decades studying the deep ocean, cataloging its strange inhabitants, but nothing prepared her for this. The creature is not just long, it is alien in a way that challenges the fundamental definition of what it means to be alive. Because this is not one animal. This is thousands, perhaps tens of thousands, and they all share the same body, the same mind, the same impossible existence. This is a siphonophore, and in April 2020, the specimen discovered off the coast of Western Australia became the longest animal ever recorded by science, measuring approximately 150 feet in length. But to call it an animal in the singular sense is to fundamentally misunderstand what makes this creature the most disturbing collaboration of life on Earth. What appears to be a single organism is actually a colony, a horror show of biological unity where thousands of individual zooids, each genetically identical, sacrifice their independence to form something greater and stranger than any single creature could ever be. Each zooid began as a separate entity, but through a process that defies our understanding of individuality, they merged, specialized, surrendered their autonomy, and became components of a living superorganism that hunts the midnight waters with tentacles trailing like threads spun from nightmares. The siphonophore is not just bizarre, it is a violation of everything evolution teaches us about survival. In nature, the individual fights for itself. Competition drives speciation. Self-preservation is the first law of life. And yet here, in the crushing darkness of the deep sea, thousands of individuals voluntarily erase themselves, transforming into organs of a larger being that none of them could comprehend alone. It is as if every cell in your body gained consciousness and then chose, deliberately, to remain part of you rather than swim away and live independently. The implications are staggering, the horror is profound, and the question that haunts every scientist who encounters these creatures is simple. How did this happen? How did evolution create something that shouldn't exist? To understand the siphonophore, we must travel back in time, far beyond the age of dinosaurs, beyond the Cambrian explosion when complex life first flowered in ancient seas, to an era more than 540 million years ago when the ancestors of these creatures first appeared in Earth's oceans. The world then was unrecognizable. No plants colonized the land. No animals walked on legs. The continents were arranged in configurations that would seem alien to modern eyes. And in the oceans, strange quilted organisms pressed themselves against the seafloor, filter feeding in a world where predation had not yet become the dominant survival strategy. Among these primitive life forms were the cnidarians, a phylum that would eventually give rise to jellyfish, corals, sea anemones, and the siphonophores. The cnidarian phylum is thought to be approximately 580 million years old, making them older than arthropods, chordates, and most other major animal groups. These early cnidarians possessed something revolutionary: nematocysts, microscopic harpoons coiled inside specialized cells that could explode outward with incredible force, injecting venom into anything that touched them. This weaponry changed everything. Suddenly, sessile organisms could defend themselves. Floating creatures could hunt actively. And the ocean, which had been relatively peaceful, became a battlefield. But the early cnidarians were simple. They existed as individual polyps anchored to rocks, or as solitary jellyfish pulsing through the currents. Then, at some point in their evolutionary history, something extraordinary happened, a quantum leap in biological organization. Some ancestral cnidarian discovered that copies of itself, rather than drifting apart after budding asexually, could remain connected. This was not merely a physical connection. The zooids began to specialize, to differentiate, to surrender individual functions in exchange for becoming part of something larger. Some zooids dedicated themselves entirely to feeding, their tentacles growing longer and more deadly. Others transformed into swimming bells, pulsing rhythmically to propel the colony through water. Still others became reproductive organs, gas-filled floats, or defensive shields. This transition from individual to colony represents one of the most profound mysteries in evolutionary biology. Despite having evolved over hundreds of millions of years, siphonophores have left no fossil record. Their gelatinous bodies, held together by water pressure and containing no hard structures, simply do not preserve in stone. We know they exist because we find them alive today, drifting through every ocean on Earth. But their evolutionary journey is written in living tissue rather than ancient rock. Scientists can only speculate about the selective pressures that drove this transformation. Perhaps it began in nutrient-poor waters, where individual survival was almost impossible, and only through cooperation could enough food be captured to sustain life. Perhaps it emerged as a defense mechanism, with colonies growing large enough to discourage predators. Or perhaps it was simply an accident, a mutation that prevented budding zooids from separating completely. And this accident proved so successful that natural selection amplified it across millions of generations. Whatever the cause, the result is extraordinary. Modern siphonophores exhibit three basic body plans, each representing a different solution to the problem of colonial existence. Cystonects possess a gas-filled float called a pneumatophore and lack swimming bells, drifting passively with currents. Physonects have both a float and a nectosome filled with nectophores that provide jet propulsion. Chalicophorans abandoned the float entirely, using oil-filled glands for buoyancy and relying completely on swimming bells for movement. The 2020 discovery belongs to the genus Apolemia, a physonect siphonophore that combines floating and swimming capabilities to achieve its record-breaking length. But to truly grasp the horror and wonder of these creatures, we must descend into their world, into depths where sunlight becomes memory and pressure crushes anything not specifically evolved to withstand it. Giant siphonophores like Priadubia inhabit the mesopelagic to bathypelagic zones between 700 and 1,000 meters below the surface, though some species venture even deeper. At these depths, the ocean becomes an alien planet. Temperature hovers just above freezing. Bioluminescence replaces vision as the primary sense, and food, that constant driver of surface life, becomes desperately scarce. Imagine descending in a submersible, watching through the porthole as blue gives way to navy, then to black so absolute it seems to press against your eyes. Marine snow drifts past, an endless sparse blizzard of dead organisms and organic debris falling from the sunlit world above. This is the only food source for most deep-sea creatures, and it is catastrophically inadequate. Most organisms at this depth exist in a state of near starvation.
their metabolism slowed to conserve every calorie, their bodies shrunken and fragile. Evolution in the deep sea favors efficiency above all else. Every gram of tissue, every movement, every biological process must justify its energy cost or be eliminated by natural selection. And yet here, in the crushing darkness of the deep sea, thousands of individuals voluntarily erase themselves, transforming into organs of a larger being that none of them could comprehend alone. It is as if every cell in your body gained consciousness and then chose, deliberately, to remain part of you rather than swim away and live independently. The implications are staggering.
The tentacles are covered with nematocysts, microscopic capsules that fire with explosive force, embedding barbed threads into prey and injecting paralyzing venom. But siphonophores have taken this weaponry further than any other cnidarian. They possess 4 distinct types of nematocysts: heteronyms with spines on a shaft, haplonyms with open-tipped tubules, desmonyms with adhesive properties instead of spines, and rhopalonyms with wide tubules optimized for large prey. This diverse arsenal allows them to capture everything from tiny copepods to fish larger than individual zooids, securing prey so thoroughly that escape becomes impossible. Once captured, the prey is reeled toward the feeding zooids, called gastrozooids. These specialized polyps have elongated tentacles and mouths capable of consuming prey items, then distributing the nutrients through a canal system that runs the length of the entire colony. Every zooid, whether it swims, reproduces, or simply provides structure, receives its share of the meal. This communal feeding system is crucial to the colony's survival. In an environment where predators might encounter food once a week or less, the ability to share a single large catch among thousands of mouths means the difference between starvation and persistence. But the siphonophore's true genius reveals itself in how it attracts prey in the first place. Nearly all siphonophores are bioluminescent, capable of producing light through chemical reactions within their tissues. Most glow blue or green, wavelengths that travel long distances through seawater. But one species, Irena sirena, produces red bioluminescence, making it only the second marine creature ever discovered with this capability. This red light is invisible to most deep-sea creatures, whose eyes have evolved to detect only blue and green. But fish can see red, and researchers believe the red bioluminescent lures mimic the glow that comes from algae inside copepod stomachs, attracting fish that mistake the lure for easy prey. The bioluminescent organs, called tentilla, on these non-visual siphonophores emit their light in rhythmic flicking patterns that resemble the movement of zooplankton, creating an irresistible lure. Fish approach, drawn by what appears to be a meal, and swim directly into the invisible tentacle net surrounding the light source. The siphonophore does not need to see its prey. It does not need to chase. It simply glows, and death comes to those foolish enough to investigate. This sit-and-wait hunting strategy explains how siphonophores achieve their impossible lengths. A small, fast-swimming predator in the deep sea must expend enormous energy hunting scarce prey. A siphonophore expends almost nothing. It drifts, it glows, it waits. Its transparent body makes it nearly invisible. Its tentacle net spans volumes of water no single animal could patrol. And when prey arrives, the capture is automatic, triggered by simple contact rather than active pursuit. The colony's vast size is not a liability, but an asset, transforming the siphonophore into a living drift net that passively harvests whatever the deep sea provides. But there is a terrible fragility to this design. Siphonophores are held together by water pressure exceeding 460 bar, and when brought to the surface, they burst, their hydrostatic skeletons collapsing without the crushing force of the depths to maintain structural integrity. This is why specimens dredged up by fishing nets arrive as unidentifiable blobs of gelatin, their colonial architecture destroyed, their zooids scattered. For centuries, scientists examining these remains had no idea what they were looking at.
It wasn't until submersibles allowed direct observation in natural habitat that the true form of these creatures became apparent. In 1987, researchers from the Monterey Bay Aquarium Research Institute became the first to observe living Praia dubia in its natural environment during a systematic study of the water column in Monterey Bay. What they witnessed challenged assumptions about deep-sea life. The siphonophore was not a passive drifter, but an active swimmer. It used bright blue bioluminescent light to attract prey, and when finding itself in a region abundant with food, it held position and deployed its curtain of tentacles like a fisherman casting a net. The colony moved with purpose, with something approaching intelligence, though it possessed no brain, no central nervous system, only thousands of interconnected zooids somehow coordinating their actions into coherent behavior. This coordination is perhaps the most disturbing aspect of siphonophore biology. How do thousands of individual zooids, each theoretically capable of independent existence, synchronize their movements? Research shows that individual nectophores positioned at different locations along the colony serve different functions based on their developmental stage. Smaller, younger nectophores cluster near the top, controlling turning and orientation. Larger, older nectophores concentrate at the base, providing thrust propulsion. Somehow, without a brain to coordinate them, these zooids pulse in patterns that produce smooth, directed movement, adjusting the colony's position in three-dimensional space with precision that would require sophisticated computation if engineered by humans. Scientists studying this phenomenon discovered that the colonial organization itself creates emergent properties, behaviors that arise from the interaction of many simple units rather than from centralized control. This modular body plan confers evolutionary advantages, including redundancy, where even if some nectophores become damaged or compromised, others can compensate so the colony as a whole continues functioning. The siphonophore is not one mind controlling many bodies. It is many bodies that together create the illusion of one mind, a distributed intelligence that emerges from connection rather than consciousness. And here we encounter the philosophical horror at the heart of the siphonophore. Each zooid in a colony is genetically identical, produced through asexual budding from a single fertilized egg. They are clones, perfect copies, and yet they differentiate, specializing into swimming bells, feeding polyps, reproductive organs, defensive bracts. They know, somehow, which role they must fill. They sacrifice their potential for independence, for survival as individuals, and become components of something larger. Human organ systems develop through a similar process of cellular differentiation, but our cells never had the potential to be independent organisms. Siphonophore zooids do. Each one could, theoretically, break away and attempt to survive alone. And yet they don't. They remain connected, remain specialized, remain parts rather than wholes. This raises an uncomfortable question about the nature of individuality. Where does the individual end and the colony begin? Is a siphonophore one organism or thousands? When you look at your own body, you see yourself as an individual, but you are composed of trillions of cells, each one alive, each one descended from that first fertilized egg. Are you one being or a colony? The siphonophore forces us to confront the arbitrary nature of these categories, to recognize that individuality is not a fundamental property of life, but a convenient fiction we use to make sense of existence. The evolutionary path that led to siphonophores is lost to time. Despite having evolved over hundreds of millions of years, no fossil record exists to trace their development. Some paleontologists have speculated that Ventogyrus, an Ediacaran fossil from approximately 550 million years ago, might represent an early siphonophore colony, specifically the float organism of an ancient colonial cnidarian. If true, this would push siphonophore origins back to before the Cambrian explosion, making them witnesses to every mass extinction, every climate shift, every catastrophic event in Earth's history since complex life began. And they survived. While trilobites went extinct, while dinosaurs vanished, while countless lineages rose and fell, the siphonophores endured. Scientists believe they may have become trapped in particular oceanic gyral centers, large rotating current systems that have persisted for 200 million years, providing stable environments isolated from surface catastrophes. The deep ocean, with its constant temperature, stable pressure, and insulation from surface chaos, became a refuge. While the world above experienced ice ages and asteroid impacts, the siphonophores drifted in eternal darkness, their biology so finely tuned to stability that radical change became unnecessary. This stability explains their remarkable diversity, despite relatively low species numbers. According to the World Register of Marine Species, 175 valid siphonophore species are currently recognized, distributed across 16 families and 65 genera. This is surprisingly few compared to other marine groups, but each species occupies a specific ecological niche, adapted to particular depths, temperatures, and prey types. Some, like the Portuguese man-of-war, float at the surface, blown by winds. Others, like the giant Physonix, inhabit depths exceeding 2,000 meters. A few species in the Rhodaliid family have even evolved to live tethered to the seafloor on volcanic hydrothermal vents up to 1,000 meters deep, abandoning the free-floating lifestyle entirely.
The discovery of the record-breaking Apolemia specimen in 2020 was, like so many deep-sea revelations, accidental. Dr. Nerita Rilson explains that scientists on the expedition were surveying the seafloor for animals when they began ascending back to the research vessel, and someone watching the camera feed spotted the massive spiral. Word spread through the ship. Researchers abandoned their stations and crowded into the control room. Wilson later described the moment as incredible, watching this huge organism spread out like a spiral UFO hovering in the water column, something they couldn't believe they were seeing. Using the submersible's laser measurement system, they estimated the outer ring's diameter and calculated the total length. But the specimen's full extent remains uncertain. Initial estimates suggested it could measure up to 119 meters once more accurate measurements are obtained. If confirmed, this would make it not just the longest siphonophore, but the longest animal of any kind ever documented, surpassing even the bootlace worm, another ribbon-like creature that can reach similar lengths but lacks the complex colonial structure of siphonophores. The expedition that discovered this specimen was exploring submarine canyons near Ningaloo Coast off Western Australia, using the remotely operated vehicle Sebastian to investigate deep-sea areas that had never been examined before. The team was part of a broader effort to catalog biodiversity in the Indian Ocean's abyssal zones, and they discovered far more than just the giant siphonophore. They found new species of glass sponges, previously unknown hydroids, coral gardens, and at least 30 species potentially new to science. The ocean, despite centuries of exploration, remains profoundly unknown. More humans have walked on the moon than have visited the deepest trenches. We have better maps of Mars than of our own seafloor. And in this vast, unexplored territory, creatures like the giant siphonophore continue their ancient existence, largely oblivious to the surface world and the species that only recently evolved to wonder about them. Modern research on siphonophores has revealed biological innovations that seem almost impossible. Their tentilla, the tentacle's side branches that capture prey, discharge their nematocysts in perfect synchrony despite having no central nervous system to coordinate the action. High-speed video analysis shows that this strike mechanism occurs in microseconds, faster than most animals can process visual information, suggesting that the trigger is purely mechanical, activated by physical contact rather than conscious decision. The tentilla represent a complex evolutionary history of gains, losses, and convergence, with different species evolving similar structures independently in response to similar predatory challenges. Some siphonophores have developed even stranger adaptations. Certain species practice diel vertical migration, remaining in deep water during the day but rising toward the surface at night, following the nightly migration of their prey. This behavior requires extraordinary coordination among thousands of zooids,
all pulsing in synchronized patterns to propel the colony hundreds of meters upward, then returning to depth before sunrise. The energy expenditure is enormous, yet somehow the calories gained from hunting in prey-rich surface waters at night justify the investment. Others have become specialists in ways that blur the line between predation and mimicry. The cookie-cutter shark gets its name for taking circular bites from larger animals, but some siphonophores employ an equally disturbing strategy. They use counter-illumination, producing bioluminescent light on their undersides that matches the faint light filtering from above, making them invisible to predators viewing from below. This optical camouflage allows them to hang their tentacles in apparent empty water, creating the perfect ambush for prey that cannot see the threat until contact triggers the nematocyst barrage. Yet despite their success as predators, siphonophores are themselves prey. Translucent crustaceans called phronema, or prambugs, are known to burrow into siphonophore colonies, chewing their way inside and living within the gelatinous tissue, often killing the colony in the process. Turtles and large fish consume smaller species. And in the deep ocean's food web, nothing is safe. Even the giant fazan-necks, despite their size and weaponry, exist in constant vulnerability, their fragile bodies offering little defense against creatures evolved to consume gelatinous prey. This fragility connects to perhaps the most remarkable aspect of siphonophore biology: their relationship with mortality. Because individual zooids are cloned continuously, replacing those lost or damaged with genetically identical copies, siphonophores don't really die in the conventional sense unless consumed entirely. A damaged section can be regenerated. Lost zooids can be replaced. The colony persists as long as enough zooids remain to maintain critical functions. This makes them functionally immortal, barring predation or catastrophic environmental change. Some colonies might be decades old, perhaps centuries, continuously budding new zooids while old ones deteriorate and fall away, maintaining the same genetic lineage across spans of time that would encompass multiple human generations. This immortality, combined with their ancient lineage, positions siphonophores as time travelers, time fossils that carry 500 million years of evolutionary history in their transparent bodies. They are older than forests, older than insects, older than most forms of life we consider familiar. They witnessed the rise of fish, the colonization of land by plants and animals, the reign and extinction of dinosaurs, the emergence of mammals, and the recent appearance of humans. And through it all, they remained essentially unchanged. Their body plan so successful, so perfectly adapted to the stable deep ocean that natural selection found no reason to fundamentally alter it. But now, for the first time in their half-billion-year existence, siphonophores face a threat they cannot simply wait out in the deep. Climate change is altering ocean currents, temperatures, and chemistry. Acidification from absorbed carbon dioxide may affect the delicate chemical balance these creatures require. Warming surface waters could disrupt the marine snow upon which deep-sea food webs depend. Plastic pollution has reached even the deepest trenches, and microscopic particles contaminate every level of the ocean. The stable environment that allowed siphonophores to persist through 5 mass extinctions is becoming unstable for the first time in millions of years. Scientists worry that we might be discovering these creatures just in time to watch them disappear. The 2020 expedition that found the record-breaking Apolemia specimen was part of a broader effort to catalog deep-sea biodiversity before it's too late, to document species that might vanish before we fully understand them. Every submersible dive reveals new species, new adaptations, new mysteries. But the pace of discovery is painfully slow compared to the rate of environmental change. And the deep ocean, despite its vastness, is not immune to human impact. As our submersible ascends from the depths, leaving behind the giant spiral siphonophore still hunting in eternal darkness, we carry with us more questions than answers. How many more of these creatures exist, undiscovered and undocumented? What secrets of colonial evolution do they hold? Could their model of distributed intelligence, of thousands of units cooperating without central control, inspire new approaches to robotics, artificial intelligence, or human organization? Or will they simply fade from the ocean, another casualty of the Anthropocene extinction, their half-billion-year lineage ending not from natural catastrophe, but from the accumulated impacts of a single species that learned to alter planetary systems? The Siphonophore forces us to confront uncomfortable truths about life, individuality, and survival. It shows us that cooperation can be more powerful than competition, that surrender of autonomy can lead to capabilities no individual could ever achieve alone, and that the boundaries we draw between self and other are arbitrary constructs rather than fundamental realities. It reminds us that Earth's most successful organisms are not the strongest, fastest, or smartest, but those that find stable niches and adapt perfectly to them, even if that adaptation produces forms so alien we struggle to recognize them as life at all. Somewhere in the midnight zone, 3,000 meters beneath the surface, a siphonophore stretches through the darkness. It is one organism. It is thousands. It has no brain but thinks. It has no eyes but sees through bioluminescent lures. It has no legs but moves with purpose. It is a string of horror, a colony of nightmares, a violation of everything we thought we knew about what animals can be. And it has survived longer than almost any other complex life form on Earth, outlasting empires and civilizations and entire epochs of geological time, drifting patiently in the cold darkness, glowing softly, waiting.
The ocean keeps its secrets jealously, but every now and then we steal a glimpse into the abyss, and we discover that the real horror is not that aliens exist on distant worlds, but that they've been here, all along, suspended in the waters of our own planet, invisible and incomprehensible, thriving in darkness we can barely penetrate, and pressure we cannot survive, reminding us that Earth is still wild, still unknown, still capable of producing life that should not exist but does, impossibly, beautifully, terrifyingly. The Siphonophore is not a monster. It is a masterpiece, and the deep sea, that alien world beneath the waves, continues to write its story in living flesh, one colonial nightmare at a time.